Good evening, everyone. Thank you for joining us for our webinar, Challenging the Skin Probiotic Paradigm, Translating Skin Microbiome Research into Clinical Applications. My name is Angela Wilson McDonald, and I am the Senior Director of Global Education for Crown Aesthetics. I will be moderating this evening's webinar. We're excited to share this information with you as we believe it will challenge the way you think about your skin and the microbes that live within. Before we begin, let me share just a few housekeeping items. During the webinar, all lines will be muted to avoid any background noise. If you'd like to ask a question, you may do so at any time during the presentation by typing in the question box at the bottom right side of your screen. We will be asking the most prevalent questions of Dr. Hitchcock immediately after his presentation. So let's get started. Crown Laboratories is delighted to feature Dr. Thomas Hitchcock for this installment of our medical education series. Dr. Hitchcock serves as a Chief Science Officer for Crown Laboratories and is an adjunct faculty member of the Eastern Tennessee State University Department of Biomedical Science. He was the founder of Zycrobe Therapeutics, Incorporated, and served as its Chief Executive Officer until acquisition by Crown Laboratories in 2019. Additionally, Dr. Hitchcock served as a Chief Scientific Officer at Bellis Medical, now Crown Aesthetics, from its inception in 2013 up to acquisition by Crown Laboratories. Dr. Hitchcock has more than 15 years of research experience, including basic science, proof of concept, preclinical and clinical research across several therapeutic areas such as cardiology, oncology, gene therapy, facial plastic surgery, dermatology, and aesthetic medicine. Dr. Hitchcock earned his Ph.D. in genetics from Clemson University and completed postdoctoral trainings at Duke University and Yale University in biomedical engineering, vascular biology, and therapeutics. Additionally, Dr. Hitchcock performed research at Weill Cornell Medical College, where he studied multiple myeloma genetics. Welcome, Dr. Hitchcock.
Thank you so much, Angela. It's great to be here to share some of this exciting information with you today. Some of this research is going to be looking at other people's research, but some of it's going to actually be research we've done at Crown Laboratories. It's very exciting because it's been a culmination of almost 8 years worth of research. We're not going to share it all today, but what we're going to do is show you some compelling reasons why the word probiotic has been slightly misused and how when we use it in a legitimate fashion, it actually can be pretty exciting about the applications that we're going to be bringing to the market in the not too distant future. So by starting, I'd like to talk about the word microbiome, just to give a sense of what it is, because when we talk about probiotics, we have to talk about the microbiome because they're kind of hand in hand. The microbiome is, if we look at the vernacular that's commonplace, the microorganisms in a particular environment, including the body or part of the body. They can be composed of bacteria, fungus, virus, or archaea. And these are microorganisms that exist in pretty much every part of our body, from our mouth to our feet, to our armpits, to our colon. They're everywhere. And there's even though we think certain parts of our body are sterile on the inside, there's even data to show that it's not. And we're going to look at some of that today regarding the skin. It's even evident, and it's debatable. This is actually something somewhat controversial, that some of the amniotic fluid towards the end of gestation of when we have children in utero actually have microbes within the amniotic fluid, similar to what we see on skin, such as Staphylococcus epidermidis and Cutibacterium acnes. So one of the things that I'll tell you is that if you look at the QR code on your screen, what you can do is you can hold up your phone to that QR code and you can actually go to our series, Beauty and the Bacteria, which is a wonderful series, it's educational series, that we're looking at the world of the microbiome. And so what we do is we start from Microbiome 101, talk about the nomenclature, get into how the taxonomy is used, how we name bacteria, and all the way to what that means to you, what bacteria mean to you, what probiotics mean, what postbiotics mean. And then finally, we're going to end the series this month. So we'll be doing the last 2 episodes on C. acnes, which is what we're going to talk about a little bit today as well. So when we look at the microbiome, we can see that it's different throughout the body, as we just said. But what does that matter to us? Well, why it matters is because it's not just a bunch of microbes just sitting there. These microbes are on the skin, in the gut, inside the skin, and many other organs, and they're constantly secreting metabolites. They're constantly eating certain substances and secreting certain substances. And those substances can either be what we consider pathogenic, which is what we would call endotoxins like botulinum toxin that causes botulism, or they could be super helpful, like an antioxidant or something that's nutritious, like a vitamin. So they can range in whether it's beneficial or pathogenic, but the thing is, they're sitting on our skin or they're sitting in our gut and they're secreting these all the time, all the time. So if you're concerned about your ingredients in your skincare, you should be even more interested in what the microbes that are sitting on your skin are secreting. So when we look at this, we can see that things affect not just our skin, but our microbiome regarding lifestyle, stress, age, diet, hygiene, drugs that we take or put on our skin, but also genetic predisposition. And we'll see some evidence of that as well. But this is all to say that the way that we affect our microbiome, conversely, our microbiome affects us. And our microbiome can secrete things that, although we're programmed genetically one way, they can change the way our genes are expressed, which is called epigenetics. So there's actually a change to the way our body functions simply by the presence or absence of certain microbes. So here's an example of in the gut microbiome, we can actually see changes where if you add a typical microbiota versus one that's taken from people that have Parkinson's disease, you can actually change the interaction between the microbes in the gut and the way that the brain functions and the manifestation of the disease. So in essence, if we use a typical microbiota, while some of the metabolites might contribute to some of the dysfunction from the disease state, which is genetically predisposed, if we take it away, if we cleanse the whole and sterilize the gut, it actually reduces the symptoms of the disease state. But then if we add in that same gut microbes that are isolated from somebody who has Parkinson, it can actually enhance the dysfunction, because there is an association between those types of microbes and the disease state, which is compounded by the genetic predisposition. Now that's in the gut, and we're starting there because people tend to understand the gut microbiome a little more, and that's because over the years they started studying before they started studying the skin microbiome. Additionally, when we look at the drugs that we take and when we ingest them, we can see that depending on what microbes are in the gut, you can actually change the activity of the drugs that you ingest. So there are several companies that are looking at how they can make drugs that don't get interfered, aren't interfered with based on the gut microbes that we have. So overall, when we look at a model like the gut and we see how it equates to the skin, there actually are more similarities than we probably realize. And one of them is that when we look at the gut microbiome, we know that it's not simply just the presence of any given strain or any given species of microbe in the gut. It's also the environment. And so if you look here at the top portion here, let me see if I can get my pointer for you guys. If we look at the top part here, we can see that there's a mucus layer here that's secreted by the epithelium. So what that's doing is we have a symbiotic organism here where you have the balance of the gut microbes as well as the environment that is conducive to health. So we're keeping the bad stuff out, we're keeping the good stuff in, the tight junctions in the epithelia are great. However, over time or for any one or more reasons, you can have a loss of that environment, which then can manifest itself in inflammation, leaky gut, the pathogenic microbes because you don't have the ones defending it. And it's a chicken or egg type situation because that inflammation can feed into the whole state and have a cycle of inflammation. So the real question is it the loss of the environment that leads to the disease state, or is it the presence of the microbe that leads to the disease state? And it's the same question we have for skin. And the answer is that it's all involved. It's not as simple as one or the other. So another instance of this is age. So as we age, one of the things that we notice is that there's an overall shift in the constituents of the microbiome. So while the majority of our microbiomes when we're young are two organisms of the skin, is Staphylococcus epidermidis and Cutibacterium acnes. And that might shock you for those of you who think that that means that people would have acne, because those are the two most prevalent, with Cutibacterium arguably being the most prevalent bacteria on the skin. And what happens as we age, it changes the architecture of our skin. So we produce less sebum, less oils, and other architectural change and ecological changes actually happen, which make the environment for the microbes different. And what we know is that the microbes that live on our skin or in our gut aren't necessarily there for just randomly. They're there because the conditions are primed, are ready, are perfect for them to what we call engraft. So the area has a specific situation that is conducive. And in the Beauty and the Bacteria series, I make the analogy of you wouldn't plant a banana tree in the Antarctic because just because you think it's great for the local animals like monkeys. Because the thing is, there are no monkeys in Antarctica, and it wouldn't be able to survive in Antarctica, and neither would a banana tree. And so it's the same with certain bacteria, is that they're not meant to live in certain places, nor could they live in certain places, nor do they really have a benefit in certain places. So let's look at what we know about the number of publications regarding probiotics. So now that we know a little bit about how the gut and the skin have these situations where it's not just the species or strain of microbe, it's the environment, it's the genetics, et cetera. Let's talk about probiotics. It's become a hot topic. But why is it a hot topic? And part of the reason why it's a hot topic is because there's a lot of misinformation about what exactly a probiotic is. So if we look at the nomenclature, a probiotic, if you talk about like the common vernacular, like Merriam-Webster's dictionary here, we see that a probiotic is or is defined by them as a microorganism such as lactobacillus, that when consumed as in food or in dietary supplement maintains or restores beneficial bacteria to the digestive tract. Now, the problem with this definition is that it's very GI or gut specific. The problem with that is that you're now saying that in order for it to be a probiotic, it cannot be on the skin. Therefore, it's ruling out that there's any such thing as a skin probiotic. This is not true when we look at the actual scientific definition of what a probiotic should be. I'm going to skip this. So when we look at even the skincare industry, this is a coalition, a cooperation of groups that are trying to actually bring some order to these terminologies when it comes to the microbiome in skincare, in the industry of skincare. And as we all know, there are some companies that have been sued and class action lawsuits because they've used words like probiotic, but they don't contain anything living, so they consider that false or misleading advertising. So this group is admirably trying to bring some sense to it, but there's some problems because when we look at the terms that they're using, the definition that they're using. For probiotics, it says live or dormant microorganisms, and there's some examples. What's the problem with this? Well, the problem is that while that is part of the definition, it's not all of it, because they give examples of Lactobacillus acidophilus. So that may be true in the gut, but a probiotic for skin, could it really be Lactobacillus acidophilus? If we use this definition, yes, because it is a live bacteria, it is a live organism. The problem, though, is that so is, botulism, botulinum, the bacteria that produces botulinum toxin. So is flesh-eating bacteria or flesh-eating viruses. These are also microbes that are living. So would we consider those probiotic? The answer is most likely no. No, the answer is no. And so, well then, how do we bring, a true definition here? What additionally do we have to have? So one of the things that we saw from the previous definition is that it needs to be not just live, but it also has to be beneficial. So not just for the gut, but for anywhere that you're trying to intentionally use a microbe as a probiotic. So when we look at the World Health Organization through this guidelines that they produced a while ago, they basically call a probiotic a live microorganism which, when administered in adequate amounts, confers a health benefit on the host. So they're basically also saying that it's not simply the presence, it has to be in adequate amounts in order to confer a benefit. So that is more specific, but it still doesn't go quite far enough. So when we look at the way that probiotics, and let's make this full screen, when we look at the way that probiotics affect our bodies, we can see that there's ways in which it can modulate our immune system. It can normalize our gut simply by modulating the number or types of microbes outside of the probiotics themselves. and it can change our metabolism. This is mostly done on the ends here by epigenetics. So it can actually change the way that our own genetics works, our own cells work by producing signals to change upregulate or downregulate genes that affect the overall body. And that's why certain probiotics can have things like calming effects on the gut, and it should do similar things on the skin. Why, though? It's because likely the best way for you to get these types of effects is to use a microbe that is intended to be in the area that you're expecting change. So when we look at the actual scientific rationale for what should be a probiotic, we lean on the scientific literature. And in this publication in 2015, the authors stated that the definition of a legitimate probiotic should be that it displays high resistance to stressors specific to the location it's meant to be used in. So if it's on the skin, it needs to be able to thrive on the skin. If it's in the gut, it should be able to thrive in the gut. It should have the capacity to survive in that relevant area of the body. It should lack any transferable antibiotic-resistant gene. It should be able to confer clear benefits through modulation of the resident microbiome. In essence, it should be able to, one of the benefits that it should convey, or that it should produce, is actually keep some of the baddies away, while making sure that the host of microbiome residents that are able to survive are actually bringing us benefit. And then the last part is that it needs to be non-pathogenic, non-toxic, and provide protection against disease-causing microorganisms. So all this together, back to one, is allowing us to have a microbe that is well-rounded in the sense that it's not just living, and it doesn't and it's not that it gives a good benefit, but that it's able to actually engraft onto our skin, and it's able to actually thrive there to convey the benefit all the time, to holistically, basically what we call biome care philosophy. So when we look at what kind of the skincare industry provides us right now, there's kind of a dearth of this, because a lot of it, as we mentioned, there's been lawsuits because a lot of the people are using not just the wrong strains, arguably, but using wrong definitions. They're calling things probiotics when they're actually postbiotics, or the ferment products of bacteria or other microorganisms, which can also convey benefit. So we're not saying that you shouldn't use lactobacillus strains, ferment products. They can still be just as good as any ingredient in skincare. The issue is, if we're thinking about our skin and what lives on there and what's constantly secreting substances, a lactobacillus is not likely to change that all that much, because once it gets on the skin, even if it's living, it's not necessarily going to thrive there and be able to engraft. What we do know is there's certain areas of the body that lactobacillus does engraft, such as in the vaginal canal, in the gut, and depending on the strains, of course, or the species. But we do know that lactobacillus has been, when we see engraftment on the skin overall, tends to be more associated with disease states. We can talk more about that in another webinar about all these considerations and nuances. But that being said, we have to meet all of these criteria for it to be a legitimate skin probiotic that's going to convey benefit long term and not just be another ingredient of skincare, which is not a bad thing to be. But when we're talking about like a biome care philosophy, an overall holistic philosophy of just making skin better, we have to think it needs to be something that actually comes from the community and is able to thrive in the community in which it's meant to engraft. So when we look at the skin microbiome composition, there's definitely areas that the areas of the skin are very different from one another as to composition. So all these colors are different species of bacteria. So we have what's called alpha diversity and beta diversity. So alpha diversity is the diversity or the number of types of microbes in any given area. So the more microbes there are, the more alpha diversity there is. The beta diversity is actually the difference or the diversity from site to site. So how diverse from sample to sample, so either between individuals or between sites on the body. Now, the reason why we're talking about this diversity term is because a lot of people, especially in like beauty magazines and such, have latched on to this concept that more diversity equals better when it comes to the skin microbiome, because that's something that's thought about as the gut when we talk about the gut microbiome. However, it's not necessarily always the case. So an example of this is that it is better when we have more diversity for things like atopic dermatitis, where more diversity on the surface is associated with healthy skin, where less diversity is associated with atopic skin. However, remember, there are genetic and other considerations when we're talking about eczema or atopic dermatitis. However, when we think about the homeostasis of the skin as a whole, even with normal skin, we ask ourselves the question, is diversity always better? More diversity, is it always better? The answer to that is yes and no, depending on where you look. So if we look on the surface, that's a very different story than on the subsurface or within the follicles or even below the skin. So to kind of get more clarity on that, when we look at skin as a whole, this study by Rich Gallo, they basically scrubbed the surface of the skin and then took samples and did 16S sequencing to look at the amount of bacteria that they were finding in these layers below the surface of the skin. And if you look on the right side here, where my pointer is, full screen please, you can see that the bacteria that was found here was pretty consistent from top to bottom. You can see that while there's, and if you look at the scale here, you can see that while it's not extremely high, not what you would see necessarily on the surface, which would have millions of bacteria on the surface, you can see that there definitely is bacteria below the surface of the skin. But when we look at the follicle, we can see that because they have an avenue to get into the skin through the follicle or other structures, we can see that there's orders of magnitude higher amounts of bacteria below the skin. Now, interestingly, the deeper we go, it seems like the more that we see. So why is this important? This is important because when we talk about the microbiome as a whole, part of what we talk about, which we've mentioned earlier, is the epigenetic effects that the microbes have on our skin cells. And that's not just skin cells, but also the immune cells of the skin. And that is where we kind of come into this whole point of the immune cells that surround the follicle are quite plentiful. You can even argue that given the amount of immune cells on the skin, or within the skin, that it is the largest immune organ of the body. Now, we won't debate that today. We'll say that it is one of the largest, because that is indeed pretty true. That is true, and it's pretty undeniable. However, what we will say is, why is that important? That's important because if all this bacteria is prevalent in the depths of the skin in the follicle, and let's say there's an equal amount on the surface, if that is indeed true, then we have to think about, okay, well then, why? What is more important, the surface bacteria or the follicle bacteria? And I'm not saying one is more important than the other, but what I will say is that when we look at the surface of the skin, we can measure that as about 2 meters squared, and that's pretty much agreed upon. But then Dr. Rich Gallo actually went through the effort of postulating that if we look at the surface area, including the surface area of the follicles, we actually have on order of a magnitude more surface area in the follicles than we do on the surface of the skin. That being said, we have to say that the amount of bacteria or the types of bacteria that live in the follicle become that much more important because they are the ones that are going to have the most effect or the most access to the majority of the immune cells of the skin because of the huge amount of surface area within the follicles. So then the question is, okay, well then what are the bacteria that live on the surface and what lives in the hair follicles? Well, what lives on the surface is a myriad of things. We have things that are transient in nature that fall on and come off. They don't really engraft. But the majority of things, as we mentioned before, on the surface are Staphylococcus epidermidis, a small amount of others, some corneoforms, and then we have Cutibacterium acnes, which is probably roughly at least 20% on the surface, if not more. But then what lives below the surface, what lives in the follicles, and that is Cutibacterium acnes, which is also formally named Propionibacterium acnes, also known as P. acnes. Back to one. Now, you might ask yourself, okay, well, you're telling me now that a lot of what lives under the surface of the skin is C. acnes, formerly P. acnes, and I believe I've read and heard for many, many years that causes acne. So if it's so prevalent, everybody should have acne, and so you might be having a hard time believing that. Well, let's take a look at some research that we did not do, so you don't have to take my word for it. Although in a bit, we're going to show you some of our own research. And what this group saw, is that if you look at the follicles of both acne individuals and normative individuals, we can go to full screen. what you can see is that this yellow color here is actually all P. acnes, or now C. acnes. So they're all C. acnes. And we can see that in both the acne individuals as well as the normal individuals, it's pretty prevalent. If you look at the normal individuals, altogether, the average is about 89% of the follicular microbiome is C. acnes. And you can see in a lot of these individuals, it's getting close to 100%. But that's not novel, because if we look over at the acne individuals, some people have 100% C. acnes as well, and some have much less. And if you look closely, you can see that overall, there's less C. acnes overall in the acne individuals than the normal individuals. Back to one. And so this is important because it tells us two things. It tells us, A, the vast majority of what's in the follicles, the sebaceous follicles especially, is C. acnes. It tells us also that the less C. acnes is more associated with disease. And what we talked about before about diversity, that means that more diversity within the follicle is more associated with disease, not health. Interesting, because it's counterintuitive to what the mainstream kind of talking point is about diversity. So diversity is relative as far as whether it's healthy or associated with pathogenicity. And this is not the only study that has said this. If we look at this study in particular, which is an older study but also corroborates that, we see that the bacteria that are in the healthy follicles is more Cutibacterium, or is exclusively, in this case, Cutibacterium. But if we look at the next slide here, we see that we also have the influx in the acne individuals of Staphylococcus epidermidis mainly, but also a few others as well. But we have less C. acnes in these individuals and more Staph epidermidis, which is more of a surface bacteria. So what we're trying to convey here, back to one, is that it's an interesting phenomena, because not only does more diversity equal more prevalence of acne, but we're seeing something that is considered another skin commensal, Staph epidermidis, being involved in acne. So are we saying that it's what causes acne now? No, that's not what we're saying. But what we are saying is that there is a connection. And so let's think about what that connection is. So we're going to do a poll right now. We're going to give you 15 seconds to answer. And the question is, so does C. acnes cause acne or not? So go ahead and put either 1 for yes or 2 for no. It should show up on your screen. And then in 15 seconds, we'll give what was the answer here. Okay, so what the audience chose was 60% said, yes, it causes acne, and 40% said, no, it doesn't. So who's right? Let's talk about it. But while we're talking about it, let's think, is it C. acnes that causes acne? So is it C. acnes, and does it cause acne? So there's a whole debate about causation versus correlation that we won't get into, but it's the idea that just because a bacteria or a microbe is there doesn't mean it caused a disease. It could mean that the disease state caused an environment that was conducive to engraftment of that microbe. But it could be one or other, it could be both. So let's think about that. When the World Health Organization, in the same guidelines that we defined probiotics earlier, they also said in that same statement that it was recognized that it is necessary to know the genus and species of the probiotic strain, which is correct, and we mostly do. We hear about Lactobacillus acidophilus or KCI. But they also add the second caveat here, which is the current state of evidence suggests that probiotic effects are strain specific, which means that its species contains many different strains. There are subspecies, there are differences within a species, and they're saying here that it doesn't mean that the benefit is a whole species. It could be one strain or more of a given species, but it's very specific to strains. That's why some companies in the gut probiotics patent certain strains or try to make it proprietary. It doesn't really help when you can just go culture it and grow it, but it's because not every lactobacillus species is going to be beneficial for every reason, and also not every strain within a species will be beneficial. So now we're talking about the skin, so the same is true for C. acnes. When we look at C. acnes, there's hundreds, if not thousands, of strains of C. acnes. And these are in this study that was done here, not by us, by Fitzgibbons et al., They looked at the 10 most prevalent ribotypes, which is basically saying they use the ribosomes to tell us how they're associated together by sequencing them, by 16S sequencing them. And so what we can see here, if we look here at the number of clones, and then we look at the percentage in acne patients versus the percentage in normal individuals, that tells us that each of these ribotypes, whether they're associated with acne or normative skin. And so you can see some of them, like these, the RT5, RT7, 8, 9, and 10, are all very, very much associated with acneic skin. However, there's a small amount that's present in normal skin. And then we look at others where it's more like a 50-50 or an 80-20. But interestingly, there is one type here, RT6, that is almost exclusively associated with normative skin. So this tells us that everybody that said that C. acnes is causes acne, you're both right, because some strains actually can be contributing to the cause of acne, while some are actually not. And so, that means that it's both. And it's not just a strain. So let's get into that. So in the recent past, scientists have realized this, that these strains are very specific to the way that they affect the body. So they decided that they couldn't just have C. acnes as a species out there, because if you look, a lot of studies say C. acnes does this or C. acne does that, and they don't put the caveat that this strain of C. acnes does this or this strain of C. acnes does that. Now they're doing that more so. But the problem is that many people built their hypotheses and their theories based off of literature that said C. acnes does this or that. So now they had to break them up to say, okay, now we have to really look at the ribotypes, the phylotypes, and they divided them into 3 subspecies: C. acnes, subspecies acnes, defendens, and elongatum. C. acnes, subspecies acnes is mostly the type 1, which is associated very strongly with pathogenicity, where subspecies defendens is type 2, which has very low association with pathogenicity. So we, at some point, may even call these different species based on how different they are. But for now, they're just a subspecies, but very genetically different, as well as expressing different cytokines, et cetera. So when we look at acneic versus normal skin, full screen, please. What we see is that while certain groupings of different strains, so not everybody has all of one type of strain of the species of C. acnes in their skin, we can see that acneic skin, there are certain types of mixtures, like this purple strain, which represents RT4, is not as prevalent in normative skin, although it is there slightly. But there are also some types, like this one here, where it's type 1, where it's also it's RT1, that you could have the exact same composition in another individual, and that person has acne. So again, while the strains obviously can be more associated with disease state, back to 1, it doesn't necessarily mean that the strains themselves can cause the disease. They can contribute. But the fact is, you also have to be genetically predisposed, and that's also been seen by research where they've looked at the immune cells of individuals who have acne versus those who do not. And even if you use the exact same strain to cause inflammation, you can see that genetically, certain people just can produce more anti-inflammatory cytokines like IL-10, seen here on the right, and they actually produce less inflammatory cytokines like TNF-alpha and interleukin-6, as seen on the left here. So it becomes this balance change genetically caused by the strain of specific individuals. Now, it gets a little more even complicated than this. And so let's go on to the next slide, which is when we look at the interaction of the microbiome with the skin cells, with the sebaceous cells, with the inflammatory cells, it's all going to cause a different chain of reactions where they all affect one another based on how they react to any given specific strain or other cell. An example of this is when we look at the gut probiotics that are put in, they can affect the immune cells like dendritic cells or macrophages. And those can spit out certain cytokines, which then can in turn epigenetically infect the brain, the skin, or the lung. Now, that being said, what people fail to recognize is that the skin, being a huge organ and having a huge impact on immune cells as well, can conversely have an effect back onto things like the gut, the brain, and the lungs. So this means that it's important for us to care for the skin microbiome because it can have an immunomodulatory function as well. The good news is that immunomodulatory function can be positive. So if we look at this here, we see that if you have a pathogenic leaning strain of C. acnes versus a non-pathogenic or health-associated strain, it can actually affect not just the immune cells, but also the sebocytes and the keratinocytes to produce cytokines that actually push native immune cells that are trying to decide who they want to be when they grow up into one of two directions. It can become a Th17 or it can be a Th117 phenotype. That means the latter is more inflammatory, would be associated with acne formation as well as other diseases. The Th17 phenotype would be more associated with healthy skin homeostasis. So this is a great thing because it means that if we actually curate our microbiome by use of a legitimate probiotic, we could actually really move the needle on our skin health and maybe even in how good our skin looks, as well. And so if we look on the right side here, we can see up top here that pH, that's indicative of a P. acnes, now C. acnes, that's associated with, that's taken from healthy individuals or associated with health, like a type RT6. And PA on the bottom graph is that is associated with acne or disease state. And so we can see if we take the P. acnes or C. acnes from the healthy state or the healthy phylotypes, we can actually grow them in a culture, take that culture and put it onto immune cells. And these immune cells will actually then change their phenotype and start to secrete antimicrobial peptides against the bad type of bacteria, like the bad P. acnes. So that's why that graph goes down, because that shows that there's less P. acnes that's bad growing. On the second one, they did the opposite experiment where they put the acne-causing bacteria, here I'll point to it, and this is the acne-causing bacteria here, and they put that in a culture and they added that to the same experiment and showed that it has no effect on the immune cells to kill the acne-causing bacteria the same way that the good bacteria does. So what this says is it just proves this part here that the good bacteria can be immunomodulatory in a good way, back to one. So an additional way that this happens is because it's not just the way that the bacteria affects our human cells, but they can affect each other through antimicrobial peptides geared towards one another. So they call, some of these are called bacteriocins, and these are peptides which actually can affect other microbes. And so two examples that are found in C. acnes are cutamycin, which affects Staphylococcus species, and acnicin, which affects other C. acnes species. Now, why is this important? It's important because you would think that if it affects C. acnes species, it would actually kind of self-cannibalize itself. The issue is, though, it actually attacks only a particular type of other C. acnes species, the ones that don't produce the same antimicrobial peptide. And that's important because if it's the good strain, it would attack the bad strain. So in a way, acne or C. acnes can actually be self-modifying as far as the way its components are, the strains that are there. And that may be one of the reasons why when we look at acneic skin versus non, there's a pattern as to which ones are found in acneic skin versus what are not, because of this phenomena. However, things like cutamycin, which attacks Staphylococcus, might seem good on the surface because Staphylococcus aureus, MRSA, can be associated with a lot of infections that can be debilitating. I myself almost died from one back in the day, staph infection. However, staph epidermidis is one of the things that protects our skin on the surface. And so when it stays there, it's great. When it gets into the follicle, not so great. And maybe this is one of the reasons why we see acne, because of this kind of battle between trying to keep things as they should be. Now that's a lot more for another day, but these are all reasons just to talk about how bacteria can also modify other modulate the other bacteria that are on the skin. One of the reasons why a probiotic is able to convey benefit. So when we look at the C. acnes, that is, we're talking about the associations with healthy skin. One of the reasons why we have healthy skin with C. acnes is because it eats the oils of our skin, the sebum of our skin, and produces what's called short-chain fatty acids, one of which is called propionic acid, which is why originally it was called Propionibacterium acnes or P. acnes, because it produces this in copious amounts, and I mean copious amounts. It's the largest short-chain fatty acid that is produced by C. acnes. And so when we take a C. acnes ferment and put it into a petri dish with Staph epidermis strains, interesting things happen. It actually prohibits formation of biofilms from things like Staph aureus and Staph epidermidis. And here you can see this graph here. You can see that there's a decline in biofilm formation. where if we do the same thing with a control, we don't see that. And then if we take an isolate of staph epidermidis from the skin, you see the same phenomena. We can actually reduce the amount almost to zero of biofilm back to 1. So that's a great thing because biofilms could be implicated in both health, because C. acnes it establishes a niche, but disease as well, because one of the reasons atopic dermatitis is hard to treat is because the Staph aureus can actually implant itself with a biofilm to protect itself from antibiotics and other things that we use to try to get rid of it. And also we know that things like atopic dermatitis is associated with less C. acnes. So one asks, if we put more probiotically, put more C. acnes on skin like atopic, would it reduce it by destroying the biofilm and therefore allowing for a kind of terraforming or microbioforming is what I like to call it, of the skin in that area? One of the reasons why probiotic, C. acnes as a probiotic makes sense. Again, we have our own strains that we're working on, and this particular strain is XYCM42 that we've been studying only in vitro, but clinically. And we did measure here when it was being fermented, the amount of propionic acid made, and it was copious in nature. And another great reason why propionic acid is a great thing and also other short-chain fatty acids like butyric acid that's made by both C. acnes as well as staph epidermidis, is that we haven't found, but it has been found by this researcher cited below, that if you take propionic acid and you induce melanogenesis in the skin of the ears of mice, that what you can do is by applying either C. acnes or propionic acid, you can see a vast reduction in the amount of melanogenesis. What this means is sun damage, like mottled pigmentation that you see, like melasma and such, could be reduced similarly to another substance that we typically like to see in our skincare, ascorbic acid or a vitamin C derivative of sorts. The problem with vitamin C, though, is that it falls apart very quickly. It's oxidized quickly. It's not a very stable molecule. It doesn't like light, doesn't like heat. The good news is that propionic acid and C. acnes ferment in the way that it works is very stable. And there's actually another thing, because you might be saying, well, great, yeah, vitamin C, and now we know propionic acid from C. acnes, both act as a tyrosinase inhibitor. They both are able to get that skin brightening effect. Great. But you know what? I'll stick with my vitamin C because it's a potent antioxidant and it keeps me protected from UV damage and such. Well, we're going to get to that in a second. But let me first talk about a molecule that we don't want C. acnes to secrete, that it is well known for, and that is a porphyrin. So porphyrins are those little things that when you use a Woods lamp or ultraviolet light and you see those glowing dots that's associated with acneic skin, that's because the strains that are associated with acne secrete a lot of porphyrins, which are inflammatory in nature. But the thing is that what most people don't know is that the healthy associated strains like the type 2 RT6 defendens subspecies actually is not associated with a large amount of porphyrin production, as is seen on the right side of the graph here. And interestingly, as we know, when we eat like dairy products and such, if we're predisposed to acne, it tends to make us break out. Certain foods can do that. And here they supplemented the cultures with B12, vitamin B12. And in the acneic associated strains, it showed that B12 actually caused a significant increase in the porphyrins, which would be more significantly irritating. But in the health associated strains, there was no increase. And some of them don't even produce at all porphyrins. So this is another reason why it is very strain specific as to whether you consider it causing acne or not, and why you were all right in the way that you answered, although maybe not for the same reasons. So we're going to go back now to the vitamin C story, because remember, we talked about why vitamin C is something that we really, really like in our skincare. And I'm not saying it's a bad molecule. It's a great molecule. You should still use it. But what I am saying is that the proper skin probiotic, like a C. acnes, could also provide a lot of benefits similar to that. And here we see that there's not one, but multiple things that are found in the ferment product of C. acnes that are potent antioxidants. One being what is seen on the left-hand side called ROX-P. And this is actually something that's been published on several times. It's a 17 kilodalton enzyme, and it's found uniquely in C. acnes. So this is not something that's prevalent in all microbes. It's not in lactobacillus. It's not in any other microbe. It's a gene associated only with C. acnes, at least to what we know now. And this is actually a potent antioxidant that actually has been studied to be as effective antioxidant as vitamin E or vitamin C. The caveat, though, is that when we look at the ferment product of C. acnes, we know that the ferment product actually is potent for at least 24, if not more hours, which is not the same thing with vitamin C, which, like we talked about, gets oxidized very quickly. So the antioxidant power persists with the ferment products of the C. acnes, but even better, if you use it as a probiotic, it's secreting it all day long. So you're constantly making more and more of these free radical antioxidants. You're making more of the propionic acid as a tyrosinase inhibitor. You're making more of the antimicrobial peptides that are specific against pathogens. You're doing all this constitutively all day long, which is a key point of why a probiotic is going to always be better than any acute type of skincare ingredient, because it's going to perpetually be protecting you, epigenetically affecting you, versus just a transient thing that you might get that might fall apart or be washed off or whatnot. As long as it can engraft, which is one of the number one things that it has to be a legitimate probiotic. So again, really briefly here, we saw that when we fractionated the large molecules from the small in the ferment product, that we saw that the large molecules and the small molecules both had pretty much the same antioxidant properties. And so that tells us that ROX-P is great and it's doing a great job, but there are other molecules that are smaller that are also giving us some great antioxidant activity. So you're getting a lot of bang out of this probiotic type of therapy with C. acnes. And so the idea is that by applying it topically, it would compete with other pathogenic microbes. It would secrete the ferment product that has all these great properties. It would live on the surface, as well as the hair follicle, which is deep into the skin, has access to all the immune cells and skin cells and sebocytes. It would actually then eat the oils of the skin, which would then, the ferment product like propionic acid would then have other great benefits. So the way to think of it is it should compete, secrete, and eat to give us that overall probiotic benefit. Now, interestingly, there is a lot of evidence, or some evidence, several studies that we won't get into, but has shown that the C. acnes can actually instigate more lipid production in the in the sebaceous gland. And so that's interesting, because that's one of the reasons why we think that, if C. acnes is on the skin of acneic skin, why they're more oily and such. But I say also that it's anecdotally been said that people that are more oily tend to age better. And why is that? Well, because you have more oil, more stuff for your good C. acnes to eat, to make propionic acid and other goodies, which can lead to protection and overall reduction in signs of aging. Theory that we'll talk about whether it conveys to clinical benefit or applications in a second or in a bit. Here we see some of our own research at Crown Laboratories that we did looking at taking skin explants from the faces of individuals. And we took those skin explants and tried to cause inflammation, or we did cause inflammation, as you can see here, full screen please, by this PGN. So we put PGN into the skin explants and it caused inflammation, as expressed by this interleukin 6 and interleukin 8. And so what we saw was that when we added IL-10 to the mixture, we saw that, as we thought, because IL-10 is a master anti-inflammatory cytokine, it reduced relatively significantly, although in this experiment not as significantly, the amount of inflammation, which was, of course, expected. That's our positive control. But what we didn't anticipate is that we actually had two strains here that are our proprietary strains. One is XYCW1 here, and one is XYCM11. XYCW1 is the wild type of our proprietary Zycrobe strain, while M11 is a derivative organism that we actually inserted a gene for interleukin 10. So it's actually secreting active interleukin 10. The issue is here we used normative individuals, and so they didn't have any predisposition to inflammation. So what we didn't expect was that just the wild type by itself secretes enough substances that is anti-inflammatory that it actually reduces inflammation more than 100% compared to control. That's super interesting. And then unfortunately, we're not able to elucidate the change here of the derivative microbe, which was what we were expecting to actually have the bang for our buck. But interestingly, when we did another experiment, we actually observed a different phenomena, which this one, it just has the Zycrobe 1, which is the XYCM42. So this is not the wild type. This is actually a derivative of the wild type that does not secrete IL-10, but has a genetic switch put into it. And that just basically means it metabolizes the same as a wild type, but we've turned off the ability for it to divide unless we give it a specific sugar molecule that is not found in food or skin that allows it to actually turn on a switch, a genetic switch that allows it to simply divide at that point until it exhausts that supply of sugar, and then it stops dividing. So you can control it for biocontainment, for safety, all those goodies, but also because it puts it in a state where it's not using its energy to do cell division, so it's putting its energy into metabolism for all the good stuff that we've talked about. But what we observed was when we took acne patient PBMCs, which is a fraction of immune cells within the blood, versus healthy individuals, we saw the same type of effects of when we put the positive controls of interleukin 10 for the most part. But what we saw here was that the acne individual, like the experiment we saw before, it wasn't as potent because they're more predisposed to inflammation. So even adding interleukin 10 did not reduce the inflammation as much as it did in the healthy individuals. And then when we added the wild type, of course, this is the one with the genetic switch turned off, we observed that it in the healthy individual, it completely mitigated the inflammation. But in the acneic skin, it did significantly reduce inflammation, but not completely. Again, just showing that it's not just the strain, it's the genetics of the individual that's going to be contingent on how we can see the benefit of probiotics. So here, the probiotic actually did its work in both individuals, or both groups, because this is a pool of different individuals. This wasn't just one person. And what we see here is that it's still helping, but those who are predisposed to inflammation may need additional benefit as well. This we don't know because this is not a clinical application. This is an in vitro application. We'll get to the clinical part. So in essence, what I'm telling everybody on this call today is that the research we're doing at Crown has basically checked all the boxes for what a legitimate skin probiotic should be. It engrafts, it thrives on skin. It's actually one of the major components of skin. It can actually help to get rid of the bad C. acnes. It can help to modulate the immune system in order to get rid of the bad bacteria. All these things for the wild type, has shown to be accurate as far as what a true probiotic should be. Now, this is, of course, all in vitro, and so we have to ask ourselves, does this actually translate? So what we did was to start looking into this is we did make that genetic switch, strain of XYCM42, but we also not only had the probiotic, we also have the ferment product. And we can use both because just like we said with lactobacillus, the ferment product contains a lot of good substances that we can use. Not all that it wouldn't be made all the time like a probiotic, but it still has benefit. So the probiotic can latch on, engraft, and have its benefits. And of course, there's always going to be this resistance to engraftment from the commensal microbes on the skin. But the thing is, there is research, multiple studies that have shown that continual application, so being compliant with application of a probiotic skin microbiome or microorganism that can engraft like a C. acnes, actually can engraft and modulate the microbiome. So we do know it's possible. It's just the method by which we do it has to be done correctly. And then the ferment can be an adjunct to that to help, because the ferment's going to have the same conditions as is conducive to the growth. So by adding the ferment with the live probiotic, you're actually helping to microbioform the skin, in theory. And of course, here's how we have evidence that the genetic switch works. So it grows in culture for 7 days. It's not detectable. You add that genetic switch inducer, it turns it on, and it grows like crazy. Now, this is one of the things that, of course, we turned it on at 7 days for demonstration purposes. But in essence, we did the research to show that, the question was asked, well, how long does a bacteria live? That's a question that in history probably has never been answered because bacteria, they just divide. They don't have a life cycle in which they live and die. They divide and then they divide again, then they divide again, then divide again. So that's why they're so predisposed to mutation. And it's why we can get antibiotic resistance and such easily. But what we did was when we turned it off, we had asked the question, well, it's not dividing, so it got to crap out sometime, and it did at about a month and a half. So if you have engraftment with this particular strain, even if you don't turn it on, you would still have residual benefit for a good while until it either is pushed out by the oils of the skin, outcompeted by something else, or otherwise craps out because of no nutrients that needs to induce division. So this is where we ask the question, but this is all great, but can we translate the microbiome research that you're talking about into a clinical outcome? Because in the end of the day, all this is wonderful in a petri dish, but if we don't put it on our skin and see benefit, whether it's health benefit or aesthetic benefit, it really doesn't matter a whole lot, right? And that's the whole point of it, is that we can't just call something a probiotic just because it does something good in the gut, slap it on the face and expect it's going to do the same. It has to be something that's measurable clinically. If we don't have clinical evidence, it really means nothing. So what we did originally back in 2017 is I was, of course, a little nervous about this because even then it was very much discussed that C. acnes equals disease, equals acne. So I said, I'm going to be the first to put it all over my face. And I'm talking about billions on my face every day for a good while. I did it for 3 weeks. And I wanted to see, does it cause me to break out? Does it cause me to have inflammation? And all the answers to that is no, 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 no. It did not do any of that. So we then had, we did a study where we took 10 individuals that volunteered, and they also tested it. And they're all relatively normative skin, but there were some interesting things because although I was expecting it to help with the health and balance of skin, there were some things that I didn't quite expect, and here's some of them. So in this individual, we noticed that because of the ROX-P and other antioxidants, we weren't seeing very much oxidation of the sebum, which is why we see blackheads, because you have oxidation, and that actually, like a banana turning brown or apple turning brown, same with the sebum. It gets oxidized and turns black, and that's what appearance of blackheads. The copious amounts of antioxidants that we're producing by the probiotic application of C. acnes, and this was simply the probiotic, this wasn't with the ferment at the time, we saw that it was a reduction of oxidation. Additionally, if you look at the close-up here, I tried my darndest to get a clear close-up of both, but you can see macroscopically, you can see the same type of benefit. You can see that not only did it reduce oxidation, but it ate the plugs, a lot of the plugs, because that's the food source. C. acnes loves sebum. And one asks the question, if C. acnes causes disease and C. acnes eats sebum, so we should probably look to see if in these plugged pores there's actually a deficit of C. acnes. As we saw, there was less C. acnes in acneic skin, and so maybe that's one of the problems that we're seeing things like these open comedones. What we did observe here, we can't say conclusively what is being done and such, but what we observed here is that we did see a reduction in open comedones based on this particular application. Now, we don't know if that would ring true for all applications, all strains, et cetera. In this particular experiment, this is what we observed. Additionally, when we looked at the microbiome of the immediate area, we did notice a slight increase in P. acnes or C. acnes, and that was something that we attributed to, because we're placing it on every day, it's engrafting more, it makes sense it would slightly bump. Now, this was based off of swabs, and so as we talked about before, we're not representing the follicle, so this is the surface bacteria that increased. And additionally, we saw a decrease in Staph aureus, which was something that we thought might happen, and albeit it was very low to begin with. This is normalized, so they're not the same. It just means that the total amount, 100% equals 1 here. So that means that even though Staph aureus was a very small amount, it reduced slightly as well. And then all Staph species together reduced slightly, which makes sense because if P. acnes is increasing, it's taking up the niche, it's going to reduce Staph slightly. Another thing that we observed was the overall reduction in how skin was red. We saw the redness was reduced slightly. Based off what we saw in vitro, it makes sense because in vitro we were seeing reductions in gene expression of inflammation. And so this would explain this, although we don't know for fact, and we can't make any clinical claims as to that. But what we can say is what we observe is skin that appears, pores appear finer. There's not as much puffiness, there's less redness, and these are all manifestations of just calmer skin, right? So this is one particular thing. And now we started to get excited at this point because then even when we were putting on this young lady's skin she had pretty great skin to the point where I remember I could not find pores on her skin. I was looking and I was like, where are your pores? But what we can see is that although she has pretty good skin, she does have some congestion when we started the study that manifested in some bumpiness and irregularity and roughness of skin. And interestingly, if you look at that 3 weeks post, a lot of that congestion is gone. And a lot of that's probably attributed to the fact that C. acnes is more prevalent, which is going to eat a lot of the stuff that causes congestion. And plus the propionic acid, all the stuff that's secreted can actually contribute to the health and appearance of the skin here. So this is, of course, pretty anecdotal in the sense that it's 10 subjects. It wasn't regulated as far as any given demographic or whatnot. It was just kind of see safety of whether people broke out. None of them did. None of them had any safety issues. So The real question is this going to be reproducible in something that's a bigger cohort that's a little more robust? The answer is yes. And the answer is we've done that study in 130 individuals, and we saw some amazing results that we didn't even expect from the results that we just saw. Now, I don't want to burst the bubble of that clinical study that we will be presenting on March 17th. And if you hold up your phone to the screen here, you can use this QR, is it QR? This Q something code, QR code, to basically scan it, and it'll tell you exactly, or it'll help you to register for this webinar. And we're going to actually show you some amazing results for a clinical study where we used a strain of C. acnes as well as a ferment of C. acnes in a biome care technology, and the health and aesthetic benefits that we saw in facial skin of these 130 individuals of all makes and models, of all Fitzpatrick skin types. It was pretty amazing. Now, I know that that's a bit of, kind of a carrot I'm dangling in front of you, and that's intentional because we are already almost out of time. I do want to get to some questions briefly. But I do want you guys to see those results. So please register. It's through JDD that we'll be presenting this. And I will be co-moderating with Jadra Hooper, who's on our advisory board for this technology. And really quickly, I want to show you this picture because if you look really closely at her face, this is not from that study. I didn't want to, ruin it, but I do want to show you this because we saw this in a study participant today from the same strain, XYCM42, using the ferment as well as the probiotic. If you look here, we have some dyspigmentation, some hypopigmentation on our chin and cheek. It's also on our forehead as well. But just so you can see it better quickly, I want to show you here. And after 4 weeks of use, we can see that not only is that better, but we see an overall, a more consistent and even skin tone. And even if you look at the oil production here, you can see a mattifying effect. And this is not something that's unique to this patient. You're going to see that we saw this quite often in our clinical study that we'll talk about next week. So now I would invite you to look at these QR codes. If you want to hold your phone up to this one, you'll be able to follow me on Instagram at dr.t.hitchcock. That is the best way to have any type of interaction with me. I get hundreds of emails a day, so I'm not unfortunately able to answer all those emails. But on my Instagram, I at this point don't have way too many people, so I'm able to manage that more easily. And also, if you do want to talk to somebody at Crown about some of this or other questions, you can email medinfo@crownaesthetics.com. This QR code will also take you there. I'll take the pointer away just in case that causes a problem. And so you can email us there, and one of our medical affairs specialists will actually get back to you. And if they don't know the answer, somebody will. So go ahead, register for the webinars. You can turn back the video if you want to get that QR code. Take a look at Beauty and the Bacteria. You can get that at the beginning with the QR code there. And this will take you to either my Instagram or the MedInfo email address. And so now I'm joined by Angela McDonald, my friend and colleague at Crown Medical, the global director of director of global education, which means she's a bigwig and she's very important to me and to our company. We are good friends because we've worked together for almost a decade at this point. And so if we rib each other, that's why, because we're almost like siblings at this point. So Angela, were there any questions that you think we should answer quickly?
There are. Thank you, Dr. Hitchcock, and thank you for that information. Thought-provoking indeed. And we do have a few questions from our audience this evening. First of all, first question is, what led you to focus on C. acnes in your research on the skin microbiome?
That's an easy one because when I started Zycrobe, and a lot of this research started when I started Zycrobe back in 2014. The reason I chose C. acnes is because when originally it was meant to be a therapeutic, we were going to do a derivative, which the IL-10 strain we talked about was one of those things. And it was originally meant to be a therapeutic. And so I was thinking, if I'm trying to deliver a therapeutic molecule like IL-10, I don't want it to be just delivered on the surface, because as we all know, it's hard to get stuff through the stratum corneum. That's the whole purpose, is I was trying to find some way to get it deep into the skin. And so I tried to find a vehicle that could actually not just get on the surface, but get all the way through the thickness of the skin, because you want to modulate the entire skin. And C. acnes was the vehicle, because it lives not on the surface only, but all throughout the follicles, and it's the majority of the follicle, as we saw, microbiota. Then I started to learn about all the benefits that C. acnes by itself has, and that's what led to this being, kind of in my mind, almost the perfect probiotic for skin.
Very nice. A lot of good information on C. acnes this evening. So, based on kind of what you shared tonight, were you telling us that C. acnes actually has anti-aging properties?
That's what everybody wants to know because, for me, like I said, it was all about just healthy skin. I wanted to increase the symbiosis of skin, get rid of dysbiosis. Anti-aging effects, of course, we all want to look better, but that wasn't my main focus. But what we were finding in our clinical studies is that the answer is yes. Vitamin C, of course, is a popular anti-aging, I guess you would call it an anti-aging molecule. And if we're seeing similar effects to vitamin C, that gets people excited. Now, what I'll tell you is when you see the March 17th clinical study results, I'll let you decide for yourself whether you think that's anti-aging. I will tell you that James Layden, who's a physician who's been practicing and doing research in this field for over 50 years, saw some of these before and afters and said, nothing on the market other than one molecule that he's ever seen can get some of the benefits that we've seen. I'm not saying that's true. I'm just saying that was somebody's opinion. Maybe you guys should tune in to the next webinar so you can say for yourself whether or not that's true.
Well, you're certainly getting us excited now, Dr. Hitchcock. So, the next question is, can any skin microbe be used to benefit the skin?
That's also a great question. And the answer to that is I don't know what other skin microbes could be used to benefit the skin, but I know that not all of them can. So for instance, Staph aureus, it's very unlikely, although it could be wrong because we've been wrong about C. acnes, right? So, it could be that there are strains of Staph aureus. I know there are strains of Staph epidermidis that there are some companies looking at it for its probiotic benefits. So yes, there are going to probably be other strains, but remember, because it's so strain specific, it's going to require that companies actually invest in research and infrastructure to grow these, because the infrastructure that's available now is mainly for dairy products. And so that's why a lot of companies are just grabbing those and putting them in skincare, because they're trying to ride the wave of the probiotic labels, but they're not necessarily doing the research that's leading them to show that it truly is a legitimate skin probiotic. Not saying that those strains, ferments, aren't good for the skin as far as an ingredient, just like any other skincare ingredient. Just saying that it wouldn't be considered a probiotic. But yes, there may be some other skin microbes someday that we'll find that are better than C. acnes. Who knows?
So this was a prevalent question that we got asked in several different ways, but I think our listeners this evening are picking up on the fact that you may be saying something a little different than other probiotic products that they have seen in the skincare market before. Are they correct in that assumption?
They are correct. And like I just said, really, it's we're not saying that the other probiotic ingredients aren't good. We're not saying that they're not useful. We're saying that what they're being advertised as is not accurate. And part of that is not really the fault of necessarily the marketing teams or the companies themselves. It's the lack of regulation on what that term means, right? If there's no regulatory way to say a probiotic is this, if there's no consensus even among scientists, then it's hard for people to, if I was a marketer, and you can argue that in some capacity, some days I try to be,
try is a good
fail to be, but when we look at that, we could say, of course you're going to try and ride that wave. If it's becoming a hot commodity and I can possibly call this a probiotic, why not? But there's a fine line between doing that when it's a live bacteria, like a lactobacillus, and saying, okay, I'm going to call it a probiotic because it is in some sense, in my definition. But if you're using a ferment, it's very difficult to justify that that's a probiotic. And so that's why you're seeing lawsuits, because there's a fine line between trying to ride a wave and trying to deceive. Mm-hmm. And I don't believe that they're nefarious per se. Anybody's nefariously trying to deceive, but that's why we have to be careful about, and that's why there are regulations around cosmetics, because you do have to make claims based on fact, not hopes and dreams.
Right.
And there was one thing I wanted to say about, and it's slipped away from my grasp, so we'll just move on and maybe I'll remember it.
Well, I think, again, thought-provoking indeed, and in typical Thomas Hitchcock fashion, if you are a follower of Dr. Hitchcock, that was a very deep dive into the science. But, I don't know about our listeners, but according to the questions we got this evening, I think we're all kind of thinking on a little bit elevated level at this time and getting a little excited about what's coming on March 17th, I believe it was, Dr. Hitchcock.
It's March 17th, and I'll put up
Central Time.
If you want to put up the full screen.
So we take it you're going to reveal a little bit more about this research and what may be forthcoming?
Yes, we're going to purely discuss very, very briefly what we discussed today, And then not so briefly, we'll discuss the clinical actually application, because today was about the science behind it. Next time is going to be more about how it translates to clinic and whether we actually see the benefits, which, spoiler, yes, we do. So it's very exciting. We're all very excited to share it with you because it's one of those things where, I think it's going to take skincare as we see it today and transform it into more holistic approach, which I call the biome care philosophy, where you're not looking at just the microbiome, you're not looking at just the skin, you're looking at it all together. So I'm very excited. And, I also want to plug a little podcast, and you can bring back the back to one. But I want to plug a little podcast called the Skin Science Podcast with Dr. Thomas Hitchcock and co-host Angela Wilson McDonald. It's actually a lot of fun. We're just getting our feet under us. There's only one episode online right now. Our second one's coming out on Friday, and we're taping the third one on this Friday and the fourth one on Tuesday. So they're going to start coming out more frequently. The topics are going to be super fun and great, and there's a lot of banter because I'm going to try and bring it down a little bit as far as the technicality, keep it still skin science. I'm going to
help him do that.
And she's going to bring the consumer perspective. We also have some great guests like James Layden, Doris Day, one of my favorite people in the world, and more. So I'd say stay tuned for that and look for my Instagram if you want the links to those podcasts.
And if any of our participants this evening want to follow in particular the story on what's coming in terms of microbiome care, where are the best places to do that? I believe you said the Beauty and the Bacteria series, and where can they find that, Dr. Hitchcock?
You can go to Crown Laboratories website, and under the R&D section, there's a section on the microbiome science. It's all there. Or you can go to our Crown Laboratories YouTube page. They're all listed there as well. Or you can go to my Instagram, where I typically have posts and links to all that stuff as well.
Well, a lot of exciting things, it sounds like, on the horizon, and we thank all of our participants this evening. And on behalf of Crown Laboratories, we wish you a wonderful evening, and we look forward to seeing you on March 17th at 7 p.m. Central. Thank you all.